I'm here with Dr. Dennis, but before we start the podcast, I would just like to say that this podcast is not intended to treat, diagnose, or cure any condition, and this information should not be construed as medical advice by me or Dr. Dennis. These are opinions for informational purposes only. Now, it's going to take me a few minutes to read through his CV because it's quite long. He got a degree in mechanical engineering, a BS, from the University of Michigan. He got a master's in kinesiology, a master's in biological engineering from the University of Michigan, and then he received his PhD in bioengineering and biomedical engineering from the University of Michigan as well. That's his education. As far as his experience, he was an engineer for the Hughes Aircraft Company, an electromechanical design engineer for Electron Products, a co-founder and chief engineer of Biologic Engineering, an independent scientific and technical consultant for NASA, and he's actually a fire chief as well.
Retired now, yeah.
Oh, you're retired now? Okay.
Yeah, because of a back injury, actually. That's one of the reasons I developed the device.
Now, as far as his academic credentials, he's an associate professor of the School of Medicine at UNC-Chapel Hill, founding chair of the curriculum in Applied Sciences and Engineering at UNC-Chapel Hill, assistant professor. He was assistant professor of mechanical engineering at University of Michigan. Before that, he was an assistant professor in the biomedical engineering at the University of Michigan. He was an assistant research scientist at the Institute of Gerontology, the University of Michigan, and he was also a visiting research scientist at MIT and Harvard and the University of Alexandria in Egypt. Now, obviously, Dr. Dennis is trying to take over the world here with that CV. I had a very good experience with the device that he created, and I don't even know exactly how it works, and I didn't even study the details of it. I kind of just took an approach where I'm just going to try it, see how it works. If I ever get tired, I actually use it as a stimulant instead of caffeine or anything like that. First question is, what do you think of the quantified self movement, citizen science, self-experimentation, and the slow way that clinical medicine is practiced?
For all but the last one, I'm very much in favor of it. I think that, we need to free science from the constraints that it's under now. It's in an iron grip. And in fact, I teach a course in tissue engineering, and this morning I went on for about 40 minutes talking to my students about what the problem is in tissue engineering and why progress is being made so slowly. You look at the greatest leaps forward in medical science for the last 1,000 years, and a lot of it has to do with people who are willing to self-experiment, to, intelligently take risks, to work things out for themselves, and for things that really matter to them. And this has been true from like the first experiments in vaccination, whether or not you like vaccinations. I mean, they resulted from a lot of self-experimentation at first. The same things with just about anything that's really important. And in the last 60 years or so, the establishment, and by establishment I mean the federal government, the universities, and the practice of medicine as it's currently controlled by insurers, has taken the innovation out of medical research. And so medical technology is today a result of that, or lack of it. And I think that the answer is going to be crowdsourced research. And in fact, I'll make the first public announcement of this. I'm planning to take all of my proceeds from this and my other devices I'm developing and actually start a foundation to do exactly that, to give people the power and the right to be part of research that's really cutting edge, highly experimental, and really moving forward. Going to reach out to foundations as well as other groups that are interested in this, but really put the power in the hands of people. And you can imagine it sort of being like a Wikipedia, but for sort of a cross between the maker movement and Wikipedia, but for people who have the courage and the intelligence to really move this forward. Because frankly, It's not happening in academia. It's not happening.
100%. Yeah, and you know what? I think especially with an ICES device or whatever, this kind of technology, it's extremely important. The clinical science paradigm is a little outdated because there's so many frequencies, and in any clinical trial, there's so many variables. You have people of a certain age group, people, male, female, a different race. Everybody's got a different genetics, and
You're
if you're
totally
trying,
right.
and if you're trying to measure something with a clinical trial, it takes 5 years just to measure one group of people. Or 10 years, whatever it is.
Well, so you know that big pharma games that. They actually, what they'll do is they'll do some pilot studies and they'll say, well, we think this drug might work really well for this extremely narrow range of diseases. And this is something people don't understand about how the FDA works. But the big drug companies will say, okay, if we look at 58, 59, and 60-year-old Caucasian males who have these kinds of like previous problems, who do not have this set of an extremely narrow defined set of people, and then an extremely narrowly defined set of like a very specific type of maybe cardiovascular blockage, and they'll really put it on the head of a pin. Then they test their drug, gets FDA approved for that, never been tested on like pregnant women, older people, younger people, people of different races. So you have these FDA approved drugs that are for an extremely narrow range of people, for the sharpest, narrowest range of things, and very often they barely get approved. You see what I'm saying?
Yeah.
So that even under the very best circumstances, they kind of don't work. And this is why we end up with all these drugs and these treatments that, it's the third leading cause of death in the United States is properly prescribed, properly taken medication. The number 3 cause of death.
Very interesting. All right, so can you explain what topics of research you've been working on in the past 20 years?
Yep, absolutely.
Yeah. So you were working for NASA in 1997. What have you been? What have your interests been since NASA? Let's say.
I finished my PhD about 20 years ago, and at that time I was working in classical physiology laboratory studying muscle mechanics and muscle diseases like Duchenne muscular dystrophy and so forth. And I did my doctoral dissertation on that. And then for about 12 years or so, I did a lot of research in tissue engineering on all kinds of different tissues, from brain, cardiovascular, musculoskeletal, all that kind of stuff. And I sort of wrapped that up in 2007, 2008. While I was doing that, in the late 90s, I was doing a lot of consulting for a really wide range of automotive and aerospace and those kinds of industries. But I did do the consulting that has kind of gotten out on the internet in the last few years for NASA and pulsed magnetic fields. And so I did all that work between 1997 and pretty much wrapped up in 2001 with them, 2002. And while I was doing that, I actually had funding from DARPA, which I think most people know what DARPA is, but for people who don't, it's the Defense Advanced Research Projects Agency. They're the kind of the sinister people in some TV shows and movies and stuff, but they're actually not sinister at all. They're the scientific arm of the Department of Defense, and they exist because of Sputnik, and their mission is to make sure we never get surprised like that again. So they take everything, biological, biomedical, and so I was doing research for them and NASA. They like to stay in the background so you don't see them much, but I actually was paid by them to figure out what was going on with magnetic fields and living tissues back, in 1998, 99, 2000, 2001. And that's how we were able to do some of this research at NASA, was on that funding. And then since then, I've been, I'm still at universities, teaching and everything, but I mostly am trying to figure out how to take reasonable technologies that would otherwise never see the light of day and turn them into something really useful for people that they can experiment with. So what I'd like to start essentially is a maker movement for people doing medical technology, because it's really kind of hard to get into. So this ICES device that we're talking about today, that's one of several other things that I've been working on. One of the devices, we may have to talk about it because some of the questions I already wanted to ask relate to how the brain responds to these things. And one of the devices I've developed with a colleague is actually the best way to tell to detect very subtle changes in brain activity. So it's a completely different thing. I can send you, if you're interested in that, I'll send you links to it. It's called Cortical Metrics. So if you just go to corticalmetrics.com, you can read all about it. I founded this company, and it basically is, I think I got one here. Yeah, I can show you. It's like a computer mouse that right now we're putting, in fact, today I'm building it up on my 3D modeler to put Bluetooth in it. But basically it vibrates your two fingertips, and it sends a signal of vibration to your brain that is actually interpreted in your brain in your somatosensory cortex. And it allows you to, with extremely high precision, quantitative precision, tell how different neurotransmitters are interacting in your brain. So it bypasses, it cancels out all the peripheral stuff that everybody's been testing for 150 years. And we can actually detect changes in your brain function if you have like a tablespoon of cough syrup. So you had somebody ask about GABA function and everything. So that's a completely separate company. It's called Cortical Metrics. We just got a huge contract from the U.S. Navy to diagnose head injury. But we really think we're going to be able to detect neurodegenerative diseases like Alzheimer's probably 15 years before you'd even be able to tell yourself that there's a problem. And then you could adjust your diet or nutrition, supplements, exercise, whatever, and you can actually fix the disease. We can show you. We can show you vectors of how different. It's an 11-dimensional analysis, and we sort of have to. It's kind of complicated, but then we project it down, and we can say, hey, you're healthy. Here's a bunch of people who are unhealthy in this cloud of data. Here's healthy people, and you're somewhere in between. We can show you how you move. So that's what I've been working on, this ICES system and this cortical metrics system for the last 7 years. It's been my core research.
When will we be able to buy that device?
Well, that's what's kind of interesting. The Navy is so interested in it. They really want to have us build one that's, you can read about it. Actually, we won the NFL General Electric Head Health Challenge last year, the 2014 one, where they're trying to solve this sort of head injury problem. Everybody has it, but it really shows up in contact sports and stuff like that. So we won that, and we started really kind of gunning towards making a really inexpensive. I mean, this is biohacker stuff. I'm not interested in overcharging anybody for anything. We're talking about putting this thing is about 10,000 times more accurate than any brain scan image you can do to tell you if something's wrong with your brain. We're going to put one on everybody's desktop for like under $60.
Wow!
With open source, it's just awesome. Now, the Navy has said, okay, well, what we're going to do is the Navy and the Marine Corps agreed, and we've signed a contract. They're going to give us about $4 million to get it FDA approved on fast track in the next 2 years. So they want us to not commercialize it for at least 2 years, but to make it, just go straight for FDA approval for a head injury. So we may be doing some beta tests. Stay tuned to your blog. If we can do it, what we'll do is we'll do some beta tests. I'll send you some. And the cool thing is, you just put Google Chrome on your machine, go to our webpage, click a button, and bam, you're testing. It's really simple.
Wow. I can imagine a lot of people on my blog and Quantified Self Movement would just be interested in beta testing it. So.
Well, that's what I want you to think about now. We're not ready to do beta tests now, but I am building 400 of them for Navy evaluation, FDA testing. We have to meet field test requirements for being used in battle, like right up at what's called a forward battalion aid station. So it has to be able to work in the mud and the muck and the rain and all that kind of stuff. And that one's going to be kind of expensive, but I'm designing it so that when we're no longer constrained by military specification requirements, then it'll just, boom, drop down to be this, it's being designed to be this $55, $60 thing you just buy and you plug it in and you roll with it. So that's the thing. So I won't have beta test models for another half a year or so, but it's something you can read about.
Very interesting.
I mean, when we get a few, I'll see if we'll get, you're still interested and we will be recruiting for beta testers. We're probably going to put it with chiropractic clinics. The reason for chiropractors is they oftentimes will see the same person every week, and our test is so sensitive we can actually tell changes. We can tell changes hour by hour. We can tell changes in a person's central nervous system week by week. So we'd like to be able to track them every week for 4, 5, 6 weeks. But if you're interested, we'll just keep in touch, and then when I'm ready to beta test, we'll allocate some for that and have some of your super readers play with it. All right.
That's great. So I'll put a link to that in the notes. what I found interesting is when you started working for NASA, you were actually a very big skeptic of all of these electromagnetic technologies, and you were actually thinking that it was very sketchy, and you didn't think that they should even go ahead with it. What changed your mind?
Data. It's, science,
What kind of
right?
data?
So the story, I'll try to tell it really briefly. The story was I was doing consulting for all kinds of engineering applications, and we were doing pretty big contracts with automotive and aerospace companies. And this guy from NASA called me and said, well, we'd like to be able to grow cells in zero gravity, and we want to grow stem cells. We want to make them proliferate much faster, because this is 20 years ago, and it was kind of hard to grow stem cells. It's still hard to grow stem cells, but it was very hard to get them to grow at that point. And I said, okay, I'll try to help you. What do you want to do? And he said, oh, well, we want to use pulsed time-varying electromagnetic fields. And I said, I know a little bit about that literature, and that's just nonsense. Don't waste your time. He said, no, we'll pay it to do it. Can you figure it out? I said, I don't think it'll work. So anyway, we negotiated, and I finally said, look, okay, I'm very skeptical. You really think it should work. I really think it shouldn't work. So let's cancel the bias out. If you want to do this test, then you pay for it, and I will design and execute a very carefully controlled scientific study to remove all the bias, to make it absolutely clear. And my goal was to prove once and for all that this was complete nonsense, right? Just, people have been talking about this for a long time. Let's do a good experiment, and let's get it published. So we all agreed, okay, it is what it is. So anyway, I make this device, and it's got all these cryptic labels on it, and they got really angry at me. Well, we should know what the settings are. No, you shouldn't. This is a double-blind experiment. I've got a secret code here. So you don't know what you're doing. That's how the science works. So you can't bias the data. So they did all the biological...
Cells?
Pardon?
On animals or cells?
Just on cells. So it was done on normal human neural progenitor, NHNP cells, which is a cell line that they use at NASA for doing tests on neural cells, right? Mm-hmm. I think it's a neuroglial progenitor cell line. So it's not like neurons that you think of it, but it's all these other cells in your brain that are really important that nobody ever talks about, which are like astrocytes and stuff like that. Totally critical, but very few people know much about.
They're actually gaining traction in the research.
They totally, absolutely. But people who know about this know a lot about it, and then everybody else has never heard of them, right? It's kind of one of those things. So it's not in the popular sort of knowledge. But anyway, that's the kind of cell we were doing stuff with. So they were doing the biology. I developed all the instrumentation. Now, critically, at the beginning, I said, so what do you want to test? And they said, oh, we don't know. Just pull something out of the literature. I said, the literature is big. I knew there was at least a couple hundred papers. Turns out there was over 600. So they said, look, we don't know. Do whatever you think will work. So I designed a system that would actually break up all possible pulsed magnetic field experiments into like 6 different categories. Sine waves, triangle waves, steady magnetic fields, square waves, pulses. Those were the basic things. And we went ahead, ran the experiment, and then to make absolutely sure we weren't just sort of thinking we saw something, we actually did gene array data. This is way back at the beginning when gene arrays were still new and very expensive, but that was our main data. So they were actually looking at gene upregulation, gene downregulation, and the analysis was done completely by a third party, so there was no way for us to screw it up, right, and bias it. So, okay, I thought, great. Now, they were kind of angry at me because I wouldn't tell them what the settings were, but I said, look, just go ahead and run the experiment, and we'll see the data. So they ran the experiment, and boom, for 2 of those subsets, those square waves and neural pulses, we saw this huge change. Everything else was nothing.
Wow. And
it was a huge change you could see in a dish. You could just see, like, this has grown a lot more. This is a lot skimpier. And you could just visually see it without even a microscope, which, looking at cell cultures, is really saying something. And then we looked at the genetic data, and it was just massive. And I thought, okay, maybe we did something wrong. So I said, I don't believe it yet. Let's run the experiment again. Let's tighten it up. Let's do it again. We did it again. We got the same result. So, although I was very skeptical, the data is the data. So I was wrong. And this is what's beautiful about science. Science, when properly done, is self-correcting. Right? I mean, you can have the completely wrong idea about something, but if you design the experiment properly, it will educate you. And many great scientific advances in history were the result of some scientist thinking it was going to be one way and then doing a good experiment and having to admit, hey, I was wrong. But you're wrong in a really intelligent way. You're wrong in this way that's the scientific method. That's why it's so powerful. It doesn't matter if you start off if you're wrong. As long as you're intelligent about proving whether or not you're right or wrong, then you're cool, right? And that's what we did. Then the dark part happened, right? So the guy at NASA, the guy who ran the lab at NASA was actually an astronaut, so he's floating around in the Mir space station. He wasn't there. His technician went ahead and patented it and published it without including me or anybody else as his invention. And he didn't invent anything. He just did the cell culture. So when you look this up on the internet, it's all in one guy's name, and my name's not on it. So when that stuff all started coming out in 2003, I was like, 2002, they're like, oh, well, we want to collaborate more. And I said, thanks, but no thanks.
But they corrected that in 2007, right?
They eventually corrected that because the guy who had been on Mir space station, he's quite a famous astronaut. He's a good friend of mine too, Dave Wolf, found out about it, and he said, whoa, hold on a second, let's fix this. He's just a completely, excellent person, and he didn't realize that this had happened in his lab. So he started an investigation. He's an MD, also electrical engineer, also an astronaut, kind of, American hero kind of guy. And he had been on the Mir for actually a record-breaking time for Americans, so he was sort of unplugged for months and months and months. So when he came back, he said, yeah, we need to fix this. And he went ahead and contacted the internal legal counsel at NASA. They did a big investigation, and they determined, yeah, actually, there was some misconduct. So that's been fixed. But the interesting thing is that, like, if you look up the patents, they don't actually put my name on them. It's a footnote down at the bottom, and you have to click the link, yeah, we screwed this up. It's kind of like, the errata in a newspaper or something. They'll tell you the wrong thing on the front page with, 200-point font, and then 3 weeks later when they do a correction, it's on page 42 underneath an advertisement somewhere for a sentence. So they don't give the same press to the truth. So there was about a 4-year period there where I just wasn't really very interested in working on it. And so the other upshot of this, the sad upshot, is that all the really important engineering and technical detail that should have been in the patents and should have been in the papers that would have really enabled people to do things, it was here. They never asked me about it. The experiments were double-blind. They had no way of knowing, so they published it. And so if you actually look at those publications, they're incomplete and incorrect in a lot of ways. So when you look at all the other devices out there on the internet that say, oh, well, we have PEMF and it's based on NASA, well, it's based on incomplete, incorrect understanding of a 20-year-old experiment.
Oh, wow. And
that's the problem with it.
And
I actually try to help these people. I try to help them. I say, no, I'll help you make it right.
Right.
Even though I'm not getting paid for it, right? I'm not getting credit for it. I just don't want them to do the wrong thing.
How do you even research when there's so many frequencies? How do you research, like, which frequencies are the best? Or is that
that's an excellent question. And it's sort of like, let me sort of start off with, have you ever played the game 20 Questions? I can think of any object in the universe. And in 20 questions, if you do it really intelligently, you can tell me, oh, you're thinking of an asteroid. Or, oh, you're thinking of a hydrogen nucleus or something. So you can, if you keep dividing the universe in half, you only have to divide it in half a certain number of times. Now, in computer science, that's called a binary search. You can have 1 to 1,000, and you can guess that number correctly in 10 guesses. How do you do that? You say, is it larger than 500? No or yes. So you have to ask sort of yes or no questions, and you have to keep splitting the universe in half. The number comes out to be about 1 quadrillion, which is a billion different ways you could set your PEMF device. Different frequencies, waveforms, how you put pulses together, how they're shaped. It's about a quadrillion. Now, by comparison, there's only between maybe, it depends on who you talk to, but there's something less than 1 million different known chemicals in the universe. Wow. So there'd be like, millions of times more different PEMF waveforms than there are different chemicals. So when people say, does PEMF work or not, it's sort of as silly as asking the question, does a chemical work for something?
Right.
Which chemical? Salt, steroid, dexamethasone, right. Water, what are we talking about? So PEMF is really huge. It's way off the spectrum in terms of all these different dimensions. And it's just hard for people to wrap their head around. It's 1 million times harder to just guess the right PEMF value than it would be to win the Super Bowl lotto anywhere. because it's just that much harder. So you have to apply a scientific method that makes sense. And that's the first thing I did at NASA was break it down into 6 pieces. So, roughly speaking, I cut that down by a factor of 10. And I kept doing that, but I did it privately because I was tired of working with NASA. It's really one person. It wasn't the whole. NASA as an organization is pretty excellent, but you get a bad habit there, right? So I kept cutting it down. And over the years, people would license the technology from NASA. And then they, for like growing stem cells or making biomolecules, they'd figure out that NASA didn't know how it worked. And then they'd end up calling me. So every year I'd get a phone call, will you please consult with us? NASA said you're the guy. Okay, fine. So I was doing this research privately for a series of companies. And finally, around 2007, I started running some critical experiments on animals, which, by the way, for the record, I hate doing animal experiments. But if you're going to do them, you can get a huge amount of information, without having to kill a lot of animals or hurt anybody. If you do this science right. The problem with academic science is it's like this huge factory, and they just grind through millions of animals, and a lot of times they just don't learn anything. So I started working with companies to do that, and the interesting thing was the companies kept getting mismanaged. So I'd get I'd get an equity stake in the company, and do some consulting. They'd pay me some money. I'd get some, they'd owe me money, and the company would collapse, and then I'd end up getting their IP, right? As compensation. So this went on for a few years, and then eventually I collected back all these things that I had invented. So it's not IP that somebody else had developed, but I would do the work for a company. They would mismanage it. The technology would work pretty well. It's just that most people don't know. Most biotech companies, even with good technologies, probably over 90% of biotechnology that's really good, that works, is crashing to the ground. It crashes and burns because of mismanagement. So it's kind of a problem generated by venture capital, and it's another whole sick system that you don't see unless you're part of it. Anyway, so I started collecting this stuff together, and then I was a fireman. Actually, I'm fire chief of the district between Chapel Hill and Durham, North Carolina. So that movie, Bull Durham, where they're driving out in the countryside, I was actually fire chief of that area. And I ended up getting a really serious back injury, going to a rescue at like 1:32 in the morning, trying to put on my gear. This lady and her kids were trapped in a house, and, I was fire chief at the time, but anyway, ended up, the fire engine ended up kind of cracking into sort of a railroad track, and I ended up twisting my back, and it was never the same. And I had always had back problems. So then I sort of took this technology I had been developing for these companies for stem cells, and I was in so much pain. It had been going on for months, and I had seen all these doctors, and they kept giving me all this, codeine, and then they said, get out of here, right? We can't help you. Or they want to cut on you or something. I thought, you know what? I'm just going to give it a try. So I put it on my back, and I remember this, 2008. I remember this like crystal clarity. It was 1 o'clock in the morning. In fact, it was 1:15 a.m. I was in such terrible pain. I'm on the internet. There's got to be a solution. So I sort of jury-rigged this thing. I tape it to my lower back, and I'm sitting there 14 minutes later. I just have like the best, anybody who's been to a chiropractor, right? You have good adjustments and bad adjustments. I had like the best adjustment I've ever had in my entire life. I was seeing stars, and it was 14 minutes. I remember thinking, wow, I must have really hurt myself. And then the pain went away. So I'm like, okay, hold on a second, something's going on, right? So anyway, that was the big breakthrough. It's like, and then, then the pain would come back, and I'd have to put the thing back on, and then I'm like, well, I'm plugged into the wall, so I need to start making this thing more portable. And it started really working, and there was a time when I was in my 40s, I had to have a walker and a cane. I couldn't walk. I was in a lot of pain. Now I'm fine.
Oh, wow.
So and in fact, I've been since then I was diagnosed with spinal osteoarthritis, which I believe I have corrected. It's my opinion. I'm trying to get another set of images to show that. So I sort of optimized this portable device for like back and musculoskeletal injuries. And it's based on all of the stuff that I learned 10 years before about musculoskeletal development in tissue engineering. So when people ask me questions like, well, why did you change the frequency from what you did at NASA, to what you're using now? I'm happy to answer all those questions, but it's because it was based on what I had observed. Some of it's published and some of it's just in my head. Things I had observed doing tissue engineering with the tissues in my hands. What made them grow better? What made them grow faster? What didn't help them? And so I sort of integrated all that information and I put it into this device. And then for a couple of years, I was the only person who was using one of these in sort of like late 2000s. And if it worked well on my back, it was great. And I started using it on friends and family. And we were doing things like people didn't have to get knee replacements anymore. And we were fixing people. And then I decided a few years ago, okay, well, it's time to kind of let this out to the world. And that's a whole other story. I had a series of bad business partners who were greedy. I had to fire them.
Wow.
It started off as a different company. I had to take it back over again. There's a lot of people out there who do not want you to have access to a technology like this. And if you do, they want you to pay vast sums of money, like thousands of dollars for it. So I finally decided a couple of years ago, you know what, screw all these guys. I'm going to take it public. If I get, and I'm telling you here, I'm putting it on, this is going public. If I get pressured by big pharma or the federal government or something, I'm going to WikiLeaks the whole thing. I'm just going to put it out there, and I'm going to put it in the public domain. So you know what, because I've had many friends like who are knowledgeable say, yeah, man, you better watch out, because there are a lot of people who just imagine what would happen if, big, good, well-selling drugs just were no longer needed for certain types of pain, certain kinds of injuries. Imagine what would happen if we didn't have to have most joint surgeries. Imagine what would happen if spinal surgery basically became a medieval bad memory of the past. That's my vision. And that's what I'm working toward.
Has anyone tried to block your progress from Big Pharma, or that's just a theoretical concern?
It's theoretical, but it's because right now I'm keeping it really small, and I generally don't talk about this kind of stuff in public. And the concern that I have is that, as soon as it gets big, people in the FDA, oftentimes the decision makers have already worked at or already have a job offer from Big Pharma. And so, the FDA would be one of the, and there are books written about this, you can get them on Amazon, where the FDA really goes out of their way to stop technologies that are really good from making it to market. And they do a lot of protection for Big Pharma. So they're trying, essentially it's like it's not a monopoly, but it's a sort of like an oligopoly, right? It's a few really big players with lots of money. So if they make it so that it costs $5 billion to get a new device or, drug on the market, sure, it's them and their buddies. They sort of circulate around the same. So, I'm not really a conspiracy-minded person. I mean, around Y2K, I was like, yeah, some elevator somewhere is going to stop working. So I don't think that way a lot. But this is actually a real problem. And a lot of very kind of sober, thoughtful people that I know have told me after they sort of seen it, they're like, hey, we need to be kind of careful about this. So, yeah, it's something I think about. But rather than waiting for that to happen, what I really plan on doing is just doing it preemptively.
Right.
Is just putting it out as part of a foundation, making it a nonprofit thing, crowdsourcing the whole thing. I'm not quite able to do that right now. I'm trying to scrape together the resources. I'm actually building a brick and mortar facility so I can do this research and pay for it myself.
Wow.
That's what I'm doing now. But what you'll see, and you should expect to see this with technology, is that every year ICES technology will get better, and it will get more reliable and more effective, and it will get less expensive. That's how technologies work.
So,
right?
Yeah, so the ICES technology right now has about 6 patents, is that right? Or 5?
2 from NASA, 4 that I own,
4,
and
okay.
probably another 4 or 5 that I haven't filed yet. I'm sort of trying to decide how to do that. 4 that are filed, that are issued, and that I own.
So this, I see.
Yeah, so it's really well. And the 4 that I have, I've had them assessed independently, and the 4 that I personally own, and I have rights to the NASA ones too. In fact, I'm actually, so far as I know, the only person who has rights to them now that's still a viable company. So anything we see that's not Micropulse that makes a claim to a NASA pedigree is doing so without a license, and they're kind of basically, I guess the right word would be a pirate. But so the IP is really strong. ICES is actually one of my trademarks. So if you see that anywhere else, that's being pirated. Right. But my goal is not to, and this should be clear to anybody, right? My goal is to make the most effective technology that you could possibly have do it in a way that's absolutely honest, and make it really inexpensive. And I mean, I've been cutting the price of this thing in half, year by year by year, and it's just and making it better and better. But it's kind of a costly process, just so you guys know. I spend a few $100,000 a year on this.
Wow.
And I'm expecting to hear back from the U.S. Army. They're supposed to give me about $500,000 in a grant to do a test in Europe, in Finland, for treating spinal cord injury. They may do it, they may not. So I'm trying to get federal research funding while they're still interested. And we've gotten a lot of this for this cortical metrics thing I was telling you about. I've been to this rodeo a few times, so I know how to do it. Because the nice thing about federal money is if they actually give it to you, then you own the data, and they don't really have any kind of ownership stake in it. It's kind of doesn't give somebody else the ability to sort of drive the train off the rails. And most investors who I've talked to, and I've talked to hundreds of them, they want to grab the IP, exclude everyone else from the market, way overprice, give the crappiest product they can, and just, it's sort of like the drug companies. They don't want to cure anything. They just want you to be a lifelong customer.
Interesting.
And yeah. In fact, there's a very famous quote from the CEO of one of the big drug companies. I won't name it because I don't want to get sued. But a very big drug, one of the big five, who said, 15 years ago, our goal is to be selling our drugs the way they sell vitamins to people all the time. We don't want to cure anything. He said this at a meeting, and then people re-quoted him over and over again. Basically, we want people to be permanent, lifelong customers. We won't cure anything.
Wow.
And that's what they've been trying to do for decades.
All right. Can you tell us how the technology has improved since the NASA days, and and the details of how it improved, as far as did the frequency improve? What exactly improved over that time?
So a lot of technical improvements have been made. Part of it is that there have been a lot of new microcontrollers and semiconductor devices, specifically voltage amplifiers and what are called field effect transistors. There's actually even been improvements in capacitors, like that hold charge. So stuff that wasn't even available when NASA was around is now available, and it's inexpensive. So the thing for NASA that I built was about the size of a small microwave oven.
Oh, wow.
And it cost about $10,000, $20,000, $30,000 to make it. Now, I can, for a few $100s, I can make one that's the size of a pack of cards. So it's gotten a lot less expensive to make, and it's because I cut out all the unnecessary stuff. So technically, in terms of the hardware, it's gotten a lot better. I figured out a lot more about how the coils should be. Everybody asks me, can't you make the coils bigger? Well, that's the wrong question. Yeah, of course you make anything bigger. Right. Should you make the coils bigger? And the answer is no. And there's good physical reasons for why they're the right size. It has to do with the concentration of the magnetic field. And the way to think about it is like an acupuncture needle. Like nobody would say, well, I've got a bigger injury, so you should use a bigger needle. It's very analogous to that. It's like, no, you actually want a really fine, sharp needle. The finer and the sharper, the better, right? So you don't have to have a needle that covers the whole injury. Directly analogous to ICES doesn't have to have a coil that covers the whole injury. You're really focally treating one part of the tissue, and that changes the way that surrounding tissue acts, right? So that's an improvement in understanding. So there's sort of like three types of improvement that have happened since NASA. One is just the hardware is a lot better. A lot of it's because my designs are better. A lot of it's because new stuff's available. I can put them into these designs. A lot of it's because I'm now producing these by the thousands, so I can make them better and less expensive, right? The first ones were all hand-built, so it's expensive. Now it's much less expensive. The second level from hardware is scientific understanding improvements, which have been many. And then the third level is in the improvement of the actual protocol itself, like what are the frequencies and stuff. So our scientific understanding has improved a lot because I have run several experiments over the years outside of the university in private laboratories and contract laboratories, and I've learned what works and what doesn't. We had a tremendously helpful experiment at Texas A&M in 2007, and you can read about this on my webpage on micro-pulse.com, and it actually told me the key parameter that you have to have right, and it's not the frequency, and it has more to do with the steepness of the leading and trailing edge of a square wave. And that has to be a certain slope. It's called a slew rate, like how quickly is the magnetic field changing, and that has to be effective for a certain amount of time. And if the slope goes down too much or it's too short, even square waves don't work anymore. So it turns out that things like the frequency 10 hertz, and then the question is, well, is 11 hertz the same as 10 hertz? Well, probably. Then you have to start asking, is 15 the same as 10? Maybe. Is 20 the same as 10? Maybe not. That's less important than the slope of the edges on the square waves. And there's a really good physical reason for that. It has to do with something called Faraday induction. You can read all about that on Wikipedia. But it's the first time derivative of the magnetic field, so the slope of the magnetic field over time, is directly proportional to the induced electric field. Right. So the trick, the key of ICES, unlike other types of PEMF, unlike TENS and electrical simulators, is that ICES is patented for and is designed for conveying a rapidly changing magnetic field deeply into tissues, and then as the magnetic field changes, it actually induces an electric field around each cell. And that's what has to be tuned right. So ICES is more like a transformer that has an external coil, has empty space, and then cells. And the fluid around the cells has ions in it, so those act as the secondary coil of a transformer. But what I'm really doing is sending energy from the primary coils directly to the space around cell membranes so that the cells can interpret that as a signal they need to respond to. So you have to do some calculus to make that work. And then, which has all been published actually, one of my doctoral students published this about a year or two ago in his dissertation. I can give you a link to that too. But basically, when you do that right, then you say, okay, so if I'm able to make these electric signals go around the cells, what sort of signals would the cells likely respond to? And that kind of brings us to the third phase of what's improved, the actual stimulus protocol. And that's been designed to simulate the types of signals that are generated during early stages of development of the musculoskeletal system. So people frequently ask me, in fact, one of the questions you had earlier was, why did I include 100 hertz and this supposed 10 hertz, which is actually 2.5 hertz? But some people got that wrong. It's really 2.5 hertz and then 100 hertz in the basic systems that we now sell, the ICES system. And the answer is that there's two types of signals that you see in the musculoskeletal system during development when the tissues are regrowing so well they practically regenerate. If you cut something off, it will regenerate. And there's very little inflammation and the growth rate is tremendously fast. So if you know what those signals are in the musculoskeletal system, then you can do some calculus and make the magnetic field from ICES send those signals to the cells. And the theory being that it will tell the cells, hey, you're in this state of early development and growth. You should heal really quickly. You should regenerate. And you should have no inflammation. So when we test that, that's what we see. We see double the speed of healing in several experiments that I've done. We see a very dramatic reduction in inflammation. And it seems to work really well for a lot of different types of tissues, but it's really kind of dialed in for musculoskeletal tissue growth, development, and regeneration. Now, how in the heck would I know what those signals are? Well, that's what I studied when I was a grad student. That's what I studied in tissue engineering for like 10 years. What are the signals that you have to send, musculoskeletal tissues to get them to regenerate? And that's something I know something about. A lot of that's not published. A lot of that is just stuff you pick up in the lab. And you publish other experiments, but these are sort of things that you know as a specialist. And that's all built into the ICES system. So I oftentimes scratch my head. I'm like, these people who are sort of copying incomplete knowledge from 20 years ago, how are they making these things work? Part of it is they're sort of lucky, but part of it is, they have these big systems. You got to plug into the wall, the lights go dim and everything. So when you compare my system that you can put in your pocket with some of these other big systems that you plug into the wall, I estimate that my system is about 99.8% more efficient.
Wow. So
what I've done over the years that's really improved since NASA is that I've done experiments to keep cutting the universe in half. Remember I was telling you there's a quadrillion. I would keep saying, so if I remove this part of the signal, does it still work? Yes or no? Wow. If the answer is yes, then that means that part of the signal can go away and I don't have to pay for it with a battery. I don't have to pump that energy into a person and maybe do harm. So I kept doing that, and now we're down to a system that can run off a 9-volt battery for like a day that has more positive biological effect than a system you have to plug into the wall. And so people, as recently as this morning, are sending me emails, oh, could you make a big, powerful system that would have all these huge magnetic fields? Of course I could. That's the wrong question. Right. I could make something that's huge and inefficient. But why? Why would I make something? And the second half of their email answers the question. Yeah, because you can sell these for $5,000 and think of the margin and blah, blah, blah. I know at least 2 people who manufacture and sell these really big $20,000, $30,000 PEMF systems who have admitted to me that they actually use my device when they're hurt.
Wow.
That is not a joke. I can't name who they are, but probably the top 2 that you would find that are like $30,000 devices, when somebody in their family is actually hurt, they use our device.
Wow.
And because it works better. It just works better.
So do you think that the specifications that you found worked optimally for reducing inflammation in the muscle tissue, do you think that would work for other tissues such as the brain?
Probably, because one of the interesting things about biology is all these cells start from one cell. And biology tends to reuse the same mechanisms over and over again once it finds something that works. It's conservation. It's evolutionary conservation. It's like, hey, that molecule worked really well, so let's see if we can recast it in this other role to do something else. And we see this all the time in biology. So my hypothesis, and I'm guessing here, I do not know that this is true, but my hypothesis is that the signaling mechanism that tells tissue, musculoskeletal tissue, hey, you're growing, you're developing, you're healthy, keep the inflammation down, grow fast, regenerate if something gets cut off. A lot of people don't know this. If a little kid who's under about 2.5, 3 years old gets their finger caught in a car door and gets it cut off, and that's a terrible thing, very often it'll grow back. Oh, wow. So humans can regenerate. Yeah, talk to a plastic surgeon sometime. They'll tell you that. It's kind of gruesome, but at an early stage in our life, we actually, as humans, can regenerate complex multi-tissue parts of our body. It's just we lose that ability as we get older. So we don't need to study, salamanders and stuff. We can study humans just at an earlier point of development, right? So what are those signals? And then to answer your question directly, why would the brain respond positively to this? Why would deep internal solid organs, why would the skin, which it seems to, when we test on people or animals that have had surgeries, their skin heals twice as fast. Why would all these other, we did a study in Egypt right before they had their revolution, where we were doing it after craniofacial surgery, and then had a separate surgeon who I've never met actually assess the recovery after major craniofacial reconstructive surgery. And in every case, the person was healing twice as fast.
Oh, wow. This was a human study.
Human study. It was done in Egypt because I have a lot of friends there in the University of Alexandria School of Dentistry. So, and I had an appointment there. We did a lot of other stuff together. So getting back to your question, why should it work in other tissues? Probably because the same kind of signaling mechanisms and the same molecules are involved in a slightly different role in different tissues. That's my hypothesis. I'm actually trying to develop a scientific way to formulate that. So I very often get asked questions about, will this help my liver? Will this help my heart? Will this help my brain? And my answer is always, I don't know. I didn't optimize it for that. But it might work really well for that. And I'm happy to give you my opinion. I'm kind of like a one-man band. I'm sort of trying to figure this all out myself, which is almost ridiculous scientifically. This is why I want to crowdsource it, right? I'd love to have 100,000 people saying, I'm willing to try it, let me try it. And then I can sort of educate them on, here are the basic things you need to do good science. So if you're going to put yourself at risk, make sure you're doing something worthwhile. And if I can broadcast that to 100,000 or a million people, we could do more research that way, crowdsourced, than all the rest of the biomedical research that's going on. You just want to make sure you don't hurt people. You want to make sure people go into it understanding what they're getting themselves into. So ICES, as a device, it only puts out half the energy that a cell phone puts out. I've tried to have it tested for safety 3 different times, and testing laboratories can't even test it because they can't even test for safety for such a low energy radio frequency device. So I believe it's very likely safe. I've also used it on myself first, and I've used it on myself continuously for 5 years with no negative side effects, all upside. Because it seems to really reduce inflammation, and I'm not just saying that. I've actually measured it in a private laboratory that does inflammation studies for the pharmaceutical industry, and I've studied it there twice, and the director of the laboratory told me this is the only device that we've ever studied that has any measurable effect on inflammation. You should know that. And not only is it measurable, it's very profound and it's very quick. Happens within an hour, it starts to really knock down inflammation. Actually, probably a lot faster than that. But that was just fine a time period because we were measuring every hour for a day, for most of the day. So we know with repeated studies done by third-party independent laboratories to FDA standards, it does reduce inflammation very significantly, even on the lowest setting. Now, that being said, sometimes reducing inflammation that quickly could initially give you kind of a negative sort of a response. So say you've got a joint injury and the inflammation is helping to stabilize the joint, but it's also preventing the joint from healing. If you take the inflammation away, the joint might become less stable for a while. So people have to know going into this, okay, I'm knocking down the inflammation. Is that going to make it take longer for an infection to heal? Doesn't seem to be the case, but it might be. Is that going to make a joint less stable so that there's more possibility that I could injure myself? That's also possible. These things haven't happened, but theoretically they're possible. One thing we do see that's really interesting is when people use it for aches and pains, they'll tell me, wow, the pain in my arm went away, but now I feel all this pain everywhere else. Well, it turns out that's your brain. And the way that your brain works is that it does something called sensory inhibition. So you know how it sort of turns off your ability to feel little annoying things all day. If you get rid of a big pain source, then your brain will be more receptive to all the other things that hurt, right? So things that you wouldn't have been otherwise aware of, all of a sudden become something that you become more aware of because the main thing that you're trying to treat has been treated. So then your brain then becomes more receptive to other signals. And that's what I'm trying to tell you. And we do see that. Very often people will say after a couple of days of using it, wow, my knee pain went away, but now my hip hurts and my elbow hurts. Is this thing killing me? And I'm like, no, actually, it probably hurt all along. And then if they keep with it for another week or so, then they'll say, wow, okay, all the pain's gone, right? So, like I say, I'm all for, bringing science to the people, letting people do things for themselves. And some people are going to, make some mistakes, but as long as you're careful and paying attention, if something doesn't seem right, stop. But all that being said, I think it's great that you're doing these things and then sort of making it public, and
I'm just telling people, try it out and see what happens.
Try it out, yeah.
In my knowledge, there's pretty much no chance that anything extremely bad is going to happen if you just try it once.
Well, I used it on my head for a long time. I know that.
You've also used it on your head.
For a long time,
oh, wow.
and
Okay.
But not as long as I have on my back. I've used it for fibromyalgia on my arms, and I was able to take care of that. I had a very, very bad case of that.
Oh, wow.
I thought I was going to have a right hip replaced about 8 years ago. Not a problem. I was able to completely fix that by putting the coils across my right hip. We were able to do the same thing with my mother and many hundreds of people where they've used them for orthopedic problems and just put the device across. And then if they stick with it for 2 or 3 weeks, all of a sudden they're like, yeah, it doesn't seem to be helping. I keep saying, well, stick with it for 2 or 3 weeks. And for problems like that, it takes a while. And all of a sudden, one morning, you get a phone call or an email. Wow, all of a sudden the pain's gone. And then if you can talk people into continuing to use it when the pain is gone, which is hard to do because it's inconvenient, about 80% of the time, the problem never comes back if they keep using it for another week or two.
Oh, wow.
And so people often ask me, well, and that's based on people telling me stuff. It's about 4 out of 5 people where it's effective if they keep using it after the pain goes away, because most people are trying to get rid of the pain. Right. But I'm saying, okay, the pain's gone, the inflammation's down, now give some time for it to start healing. If they're willing to do that, then about 80% of the time you get a permanent positive outcome. For orthopedic injuries, about 13 out of every 14 people tell me it helps a great deal. And of those who it helps, and there's some number that it just doesn't help. Not one thing's going to fix everything. But of those people where it does help them for orthopedic injuries, if they stick with it after the pain goes away for an extra couple of weeks, pretty close to permanent correction. So this is the kind of advice that I give people if they want to try it. And I cannot tell you the number of people. It's thousands now who are saying, wow, okay, this is really, really helpful for things like back pain and orthopedic injuries. So I can say a lot about that because I've got a lot of anecdotal data. But then when we start talking about things like, will it change your liver enzymes? Will it change your cognition? I know a lot less.
Right.
So I tell you what I know.
Yeah, 100%.
But I just don't know as much.
Yeah.
Right?
That's it's great that it's better to say you don't know when you're not sure of something rather than just try to
Oh, totally.
Exactly.
Now all this stuff is stuff that we can figure out, and if we end up starting to do some crowdsourced experimentation, maybe we should do that in a more sort of organized way. I'm certainly going to try to do that with my foundation, and
I'll try to get as many as my bloggers.
We can be systematic about this, and then really sort of put the information together, and let people decide for themselves. I mean, we make important decisions every day that impact our health and our safety. Getting into a car, walking up and down a staircase. These things are very dangerous.
Yeah.
Deciding to take a prescription drug is very dangerous. So we assume that as adults, we can make these decisions intelligently. And so my policy is just tell people everything you know, the good and the bad, and try to put the best technology out there, and then just let people figure it out. And I think we're on I want to kind of go back to this. I think we're on the cusp of a revolution. If this is something we can actually build into the minds of people as, hey, it's time to start doing this, and you have the tools to do this, I think we can change the way that medical research is done profoundly. I mean, we all have access now to computational power, many millions of times more than the computational power they used to put a man on the moon in the 1960s. We have access to materials at a level of purity that you couldn't get 30 years ago. We have access to information that just wasn't available. And the technology, this sort of electromechanical stuff, has become available. So there's really no reason why people can't start playing around with medical devices as a hobby and really contribute.
I completely agree. Now, let's get into some of the mechanisms here. I happen to think that... So, first of all, tell me if this is right. Are there electrons being transferred?
Yeah, absolutely.
Yeah. So do you think
there's
those...
the electron transport system.
No, from the PEMF, the ICES device, are there electrons transferred, do you think, to your mitochondria?
No, I don't think so. What's being transferred is, I could be wrong,
okay, yeah.
But what I think, the way it's designed to work, and sometimes things don't work the way they're designed, right? But the way it's designed to work is by electromagnetic induction.
Oh, I see.
It's designed to work as you have a magnet, and it makes a magnetic pulse, and that magnetic pulse goes through space, and then it causes an electromagnetic field that opposes that change. So this is how maglev trains work. Wow. This is how old-fashioned E-track tapes work. This is how the induction cooktop on your stove works, but that's thousands of times more powerful. So the idea is that every cell has a sort of oligomolecular, so just a few molecules thick, layer around each cell that has ions in it. So they're not electrons necessarily. Mostly they're ions, like calcium, potassium.
Right.
And they're resident right outside the cell membrane. And then your cell does a whole lot of things by how it moves those ions across the membrane in and out of the cell. So if you think of this as a cell membrane, a lot of cell signaling has to do with how the ions go through that membrane, and they have to be close to the membrane for that to happen. So we know a lot about that kind of signaling, and that's what they use things called patch clamping and voltage-gated channels and all this stuff that we've known about for 20 or 30 or 40 years. That's how that kind of signaling works. But what has not been tested is, can we see ions moving across the membrane, either on the inside or the outside? So if the cell has a magnetic field that's being created or collapsed in this direction, the magnetic field, there will be an electromotive force, a voltage, causing ions to move in one direction or the other around the membrane. And what has never been studied is, can the cell detect that kind of ion movement? Well, it has been hypothesized. The hypothesis for this has been around. It wasn't my idea. People are trying to figure out how you can detect exercise and other things like these really tiny little signals that we know must be generated. So if you imagine your bones or your muscles as sort of like a sponge filled with water, and that water has little ions in it. When you squish the sponge a little bit, those ions have to move. So that's the equivalent of an electric current. It's a movement of charge. So if you can imagine a situation in which some charges move a little bit easier than others, which is what biology does, it controls what moves where pretty well, then you get a difference in how the charges move every time you squeeze the tissue. And that may be the signal that tissues see when they detect exercise. It is not known. Everybody's like, oh, maybe it's lactic acid, maybe it's this. Well, one of the sort of esoteric theories about it is what's called streaming currents. And what they mean by that is, it's the movement of ions that the cells can somehow detect. So if you start with the assumption that might be true, which is what I did, and then you start designing to make a system do that, it works really well. So I haven't proven that is the mechanism, but I do have 2 collaborations that are scientific with people who are very good at determining cell signaling to see if we can actually run the experiment, the series of experiments necessary to prove that that's what's happening, that's how the signal is working. And in doing that, we'll probably discover a whole new class of membrane-bound receptors for not transmembrane ion transport, but paramembranous or epimembranous ion transport, which those receptors have not been identified, they haven't been discovered, they haven't been described. But that's what I'm hypothesizing is there. Then when you design the system to tweak those, and you make that signal look like exercise, the cell responds as if it's a sort of a young child's cells responding to exercise. But the cool thing is you don't actually have the injury that goes along with the exercise. So you get just the signal that says grow and repair, but you don't get the mechanical pounding that also gives you the other side of that coin, which is the actual injury. So your cells are responding to injuries that aren't necessarily even there.
Would you know what that flow does to the mitochondria?
Oh, well, a lot of people ask me that question, but the thing you have to understand about a mitochondrion is that mitochondria are really central for electron transport.
Right.
Very important in metabolism, right? I mean, it's like 20-25% of the cell volume of your cardiac cells, cardiac myocytes, is mitochondria. It's just absolutely critical for, metabolism and ATP cycling and all this stuff, and electron transport. But a mitochondrion looks like a little bacterium that's inside your body, and it has a membrane. And so because of the way induction works, there would be a paramembranous flow around the mitochondria as well. That's as far as I'll take it. I can prove by physics that I know that that's happening, and I can actually tell you how much of that's happening. What I cannot tell you is, are the mitochondria sensing it? How are they sensing it? How are they responding? That's like an open scientific question mark that I think is a great thing to study, and I'm working on. And I'm doing it sort of privately because academia is broken, and it would take me, let me tell you, doing my research privately with my own money, if I do the same experiment at a university, it would cost between 20 and 50 times the number of dollars. Yeah, it's not a joke. So it's 20 to 50 times more efficient dollar-wise to do an experiment privately than it is in academia, which is going to change the face of academia in the next 20 or 30 years. If there's a Republican out there somewhere who's also a senator who's listening to what I'm saying right now, that may change the policy for dumping money into academia that's just getting squandered. It's something crazy like between 80 and 95% or 99% inefficient in academia to do research.
Wow.
Because you have to pay for so many other things, and it's just the whole system is built. It's this huge parasitic thing. So anyway, enough editorializing. I want to figure out how this is happening, but I don't need $100 million to figure it out. I need to spend $1 or $2 million, and I'm going to. When you buy one of these systems from me, that's what it goes for, because I use it to develop the product and do research. That's why I don't give them away for free. Of course, you're paying for hardware too and everything, but that's part of it. We want to understand what these things are. Now, you see a lot of people on the internet talking about, it tunes the mitochondria, it does this, it does that. Hey, you're talking about Dennis. I was wrong from the very beginning about this. I just don't know, right? I mean, I was the one who said this doesn't work, this is nonsense. So keep that to heart when I say they're probably full of it. All I'll say for sure is they don't show me any data to prove what they're saying. Right. So I have no way of knowing that it's true. But people will say this, and it's sort of pseudoscientific, and then they'll refer to, oh, the NASA work proved this. Okay, well, prove it to me. I was there, right? I was doing it. Prove it to me. Or they'll say, oh, well, we did a study, but we're not going to share the data. Hey, somebody wants to challenge me. I send data. Right. Here you go. Check it out for yourself, right? They're making these statements and they're making these inferences, and then they're assuming enough people will believe them to buy their product. But I really, I don't see the expenditure from any of these other companies on research. And you can prove that. Look at their devices. They basically say the same year after year after year. The price doesn't go down. The effectiveness doesn't go up. It's a static technology, so they're not developing the science, the understanding. They're not developing the system. The only thing they put money into is marketing, and that includes putting it in an extra package, maybe putting a touch screen on it. None of that stuff's going to help you.
And
what's going to help you is the core science.
It seems like your company barely puts anything on marketing. It's mainly...
We put nothing in marketing. We put zero on marketing. I spent nothing on it. I'm just a geek in a garage who does his own crazy mad science stuff. That's what I do. And let me tell you something, I happen to know how big this market is now. And, my friend, Dr. Bill Pollack, who I respect greatly, he's an MD. He's widely regarded as the world expert in pulsed electromagnetic fields. He has drpollack.com, somapulse.com, bodyfields.com. He runs this as a business. He is widely regarded as the fair broker for PEMF devices. He sells all the ones that you can sell in the United States. He's even been on Dr. Oz, I guess it's about 3 years ago now, talking about PEMF, and you can find this video. It's on the internet, and Dr. Oz concluded, this was before he started selling our device, that PEMF probably does work. It's kind of subtle, but it probably does work. The reason I'm telling you all this is that half of all the sales that Bill Pollack makes are our technology. That's half of their business. I think probably more than half of the PEMF devices sold in the United States are our devices. Based on that and some other information that I have, because I know some of the other big companies that are doing it, and they actually ask me for help, and then we get to talk about things like their businesses for the big machines are really going down. And most of them blame me for that. They're like, if you can buy something for a couple $100 that you can put in your pocket that works better than something that costs $20,000 that you got to plug into the wall, why buy the big device? So I know those guys are all uniformly afraid that their market is vanishing. And maybe it should. I don't know. I mean, I just want people to have the best thing. So the reason I'm telling you all that is that I don't have to put money into marketing. I simply try to make the absolute best technology. I don't put any extra bells and whistles. People ask me all the time, why does it look like an iPhone? Because you don't interact with it the way, an iPhone interacts with your eyes and, your thoughts and your fingers, and you're constantly exchanging information. So you have to have a touch screen and all that stuff. This device is supposed to be ideally 0-button simple. You turn it on, you put it in your pocket, and you go, and it works. So you don't need a touch screen. You don't need all of these other features. I may put Bluetooth on it so that you can set it up with your iPhone and just forget about it. But, if you wanted to have a touch screen on it, you'd have to pay for the development costs of a touch screen, which, when you're only making 1,000 units a year, you probably add $1,000 per unit to the cost. You see what I'm saying? So none of this sort of unhelpful bells and whistles. When you buy one of our products, it's just this raw technology. $0 spent on marketing. It's all done by word of mouth and by bloggers who've tried it and who use it. There's actually a lot of doctors and chiropractors who use it now too in their practice. And so it's just that kind of level of honesty that, and it's not doesn't seem to be hurting me, because I think my best information is that we have captured about half of the domestic market for PEMF devices just by having an honest, properly priced product.
Very interesting. Now let's actually, I'm going to take a little turn, but I'm going to come back. I'm just curious what you think of, and I think you would be one to know what's going on with this. There's some people who think that EMFs in general, like low-wave EMFs, ELF EMFs they're called, are one of the drivers for disease. I actually spoke to a person, a neurosurgeon, a brilliant guy. He's been studying physics, biology, chemistry. He's actually a dentist as well. and he thinks like that's like one of the biggest drivers of disease. And I mean, if you just look at some, there's been an explosion of autism and things like that since the 90s, since all these wireless technologies are coming out. Now, what do you think of EMFs as far as the? Do you know anything about the potential harms, or do you think it's quackery?
Actually, I am looking. The reason I was looking over here while you were talking, I was listening to you. I wanted to pull up a web page of a friend of mine, and her name is Dr. Magda Havas, Havas, and she is associate professor of environmental and resource studies at Trent University, which is relatively close to Toronto, I guess. And anyway, she's been here to visit me a few times. She's tried our device, and she is the world expert, widely recognized world expert in electromagnetic sensitivity syndrome.
Oh, interesting.
And she and I spoke for about 2 hours on the phone a few months ago about this. And she has grave concerns about it because she knows many, many hundreds of people who suffer terribly because it was funny because the first time she visited me, I had, my students, we were building these big Tesla coils and we were trying to transfer energy wire. I'm a mad scientist, and it just mortified her. She's like, "Oh my God, do you have any idea what you're doing?" And I said, no. Right? So anyway, she educated me on this over the last few years. I think it's absolutely true. I think people aren't sensitive. I try really hard to make this technology that I'm building as clean as possible, and that's why one of the reasons I keep driving off all the unnecessary parts of the electromagnetic signal is if I don't have to generate them, and I don't have to pay for them with a battery, I don't have to pay for them by being plugged in a wall, and I don't have to subject a person to that extra energy. It's like an impurity in a drug, right? You want your food to be pure. You want your medications to be free of impurities, and this is the fundamental problem I have with people who are not scientific about PEMFs, is that and making a product that they just want to sell and make money off of. I would rather help my competitors and help them not poison people with extra garbage in radio frequency spectrum than just sit back and let them do it. So I actually help people, and it costs me market share and money to putting my business hat on. I'm an idiot. Stop that. But the thing is that I took my inspiration from Mercedes-Benz, and I don't know if they still do this, but for many decades they would actually do all these automotive safety tests, and then they would just make the information widely publicly available so that every automaker could incorporate what they learned. And many of these safety features in your cars were basically given away for free by Mercedes-Benz because they just didn't want people to get killed in cars. They're just tired of it. The injuries and that. And I thought that was a really cool corporate decision that we just don't see enough of. So I'm committed to, if I see somebody with a product that I consider dangerous or stupid or something like that, which is anything that's overpowered, I try to help them. And some of them are just, don't respond. But some of them actually, I've become friends with these people and I'm helping them. I'm saying, your system would be more efficient and effective if you made these technical changes. It's kind of funny, but I do that. And so you see sort of like the rising tide raises all boats, right? So you see a lot of these other big expensive systems, they seem to be now lately getting a little bit better. A lot, I believe, part of that is because I'm helping them, and they're not doing as much harm because all that extra energy is going through your body and it's not helping you.
Do you try any, this is actually not related to the ICES device, but do you happen to try any mitigation, like try to turn your Wi-Fi off at night or anything like that?
Yeah, actually I do. And in fact, I even have there's gosh, I can't remember the name of the webpage now, but they make grounding equipment, so you can.
Oh, I was going to ask you about that too.
Yeah, totally. I own their silver, strand bedding, stuff like that. Part of it is I try not to pollute, right, so much into the radio frequency spectrum. It's like turn stuff off if I'm not using it. I try to make sure my products are absolutely only use the energy that you need to have and don't have anything else. And that just makes a better product, right? 100%. I mean, it's like get rid of the extra noise and the impurities and stuff. Just do the thing that works. And so as I try to hyper optimize the product, I'm really kind of focusing on the orthopedic space. And here I'm going to hypothesize again. I would have thought that it would have become less effective on other tissues like the brain, but it may be that because the signal is so purified that the negative effects on the brain or other tissues have kind of been canceled out, that you might be able to see these subtle positive effects. That's not what I would have guessed. But as people like you testing these things on yourself, using it with absolute minimum energy that's necessary to have an effect orthopedically, starting to see beneficial effects.
Yeah, actually, some other people. I mean, I got a little feedback so far, and so far the feedback has been positive. One person it didn't do anything for, but that person was actually quite resistant to a lot of, just in general, to a lot of things. But I'd say like 4 people have benefited from it so far. So I'm following up with them. I'm going to see, you know, how far they can take that.
If you're seeing something like in the 80-90% range of people saying, yeah, it's helpful, that would be very much what I'm seeing. And it's also very, very much above placebo, which would be about a third.